Hello, everyone. Thank you very much for joining the YouTube channel of Advanced Multiple Incorporation. Advanced Multiple Incorporation actually is knowledge processing and information management institution that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation, Ontario, Canada. Both of these organizations are located in Mississauga, Ontario, Canada. Today, I'm going to discuss my knowledge, my experience, and my research under the title History, Molecular Structure, and Pathophysiology of COVID-19. For this purpose, I have collected the scientific information from different data sources like PubMed, Medline, American Chemical Society, Scopus, Google Scholar, Embase, directory of Open Access Journal, ProQuest, Journal Masterlist, CNKI, that is China-based database company that provides the scientific information all over the world. Some information collected from ResearchGate and Crossref. Before moving forward, I would like to discuss a little bit about history of COVID-19. This is not a new challenge. This is not a new microorganism. This is not a new virus. This was discovered approximately 90 years before. In 1930, coronavirus was reported first time in history. In 1931, Arthur and Hahn, they were first scientists who reported corona as a new respiratory viral infection in chicken in North Dakota, USA. In 1960, human coronavirus strains were identified. It means this strain was discovered and identified, having the potential to introduce or produce infection in human population. So in 1970, Stola he confirmed that this virus may become pathogenic for human population in future. 2005, Khan reported it one more time with additional molecular feature. So ultimately, in 2019, novel coronavirus strain was discovered and reported in Wuhan, China, and afterward it is identified globally all over the world, in different part of the world, and become very big challenge for the human being. Okay, now we are going to discuss a little bit about the structural model of coronavirus based on tomogram. Tomogram is a scientific machine that is used to read the molecular or different little structure, non-visible structure of different microorganisms. So it have very little invisible segments or the components called as ribonuclease protein complex, envelope protein E, membrane glycoprotein M, and spike protein S. Spike protein are most popular part of the virus because most of the pharmaceutical research institutions they have targeted this particular part of the virus, and they produce, synthesize, or develop the antibody against this antigen. This part of the virus is considered as antigen, and they have tried to develop or they produce the antibody against this segment of the virus. When it is damaged, it means you have damaged the virus. The virus is damaged, it means you have controlled the infection. Okay, well, the molecular characterization of COVID-19, the term coronavirus refers to prominent crown protruding spike. This is RNA virus enveloped helical nucleocapsid along with positive polarity. Picornavirus, Hippivirus, Calcivirus, Flavivirus, Progavirus, Retrovirus, and coronavirus belong to same family. They have approximately same positive polarity in RNA, whereas other part of this family having negative polarity. It is single strand, linear, and non-segmented genome. If we talk about the symptom or the syndrome induced in the patient, we find that like major symptom that are developed in human patient are fever, fatigue, dry cough. The other syndrome we see in patients are body ache, pain, runny nose, sore throat, shortness of breathing, and diarrhea. Now we are going to discuss a little bit about the transmission and life cycle of this virus. Whenever it enters into our respiratory system, that is more likely route of transmission of the virus. It is airborne microorganism. It is transmitted through the uh, respiratory droplets. Whenever it enters into the respiratory system, it attaches with the alveoli. In which part of the alveoli? Alveolar cells. Within the alveolar cell, it attaches with the ACE receptor. This is a particular place where this virus get attached and find the door or the gate, enter into the cell. ACA is angiotensin converting enzyme. Whenever it enter into the cytoplasm, it produces the vesicle to protect himself from the cytotoxic material, from the host defense. Whenever it is protected, then eventually it is released into the cytoplasm itself, become messenger RNA. It means it go through the ribosome and start producing the proteins. These proteins then assemble, they get together to produce the progeny. Progeny means the baby virions. These baby virions then develop in sufficient quantity. They burst the cell. When they burst the cell, it means they destroy and damage the tissues, and they distribute it to the whole body and induce the infection. When they distribute it into the whole body, it means they produce the symptom of the viral infection, COVID-19. Whenever they produce the symptom of the viral infection. On other hand, these viruses also captured by the antigen presenting cells, macrophage. These antigen presenting cell process the antigen. Antigen means virus. They convert it into the viral epitope. These viral epitope presented to the T cells. T cells are the component or the part of the immunity. Whenever T cell capture it, they identify it, then produce particular leukotrienes. Leukotriene is a mediators, that is stimulating material, that is a like a chemical that is synthesized by the T helper cells. These leukotrienes then activate the B cells and cytotoxic T cells. When these two cytotoxic T cells and B cells are the component of our natural defense or immunity. B cell then start producing the antibody. When antibody produced, it means the person become immune and he get the immunity within the body. Whenever he get again sick with the same organism, the antibody, they attack on the antigen and kill it, destroy it, and protect the person. Now we are going to discuss a little bit about the pathophysiology of COVID-19. When we talk about the pathophysiology of COVID-19, then we compare like a healthy person, healthy organism, healthy cell, or healthy tissues with the sick person, sick cell, sick tissue. So we have four different of the model, four different of the prototype alveoli. Alveoli are the functional unit of our respiratory system. The first one is healthy, second one is infected, third one is moderate, and last one is the severely moderate, where patient cannot survive. He most of the time expired. In if we say that a patient is infected, it means there is inflammatory material. If we say that there is inflammatory material, it means there is cytokine, there is cytotoxic material, maybe histamine, maybe prostaglandin, or maybe leukotriene. These cytotoxic inflammatory material then accumulated within the alveoli, and they hurt or they damage the normal functionality of that particular cell, particular tissue, particular organ. When we say that it stops the functionality of that organ, it means our respiratory system, they stop exchanging the carbon dioxide and oxygen. Our RBCs, red blood cells, they may not able to capture the carbon dioxide from the alveoli, and they may not able to deliver the carbon dioxide at alveoli, and that eventually exhale from the body. So our respiratory system is damaged. When the respiratory system don't work properly, it means we don't have oxygen in the body, and patient may not able to survive anymore. So in conclusion, the COVID-19 came as a serious challenge, posed potential threat, and induced serious damage all over the world. The availability of the epidemiological features, phylogeographic data, and phylodynamic information helped the virologists and scientists to develop the vaccine in shortest possible recorded time in human history. The genome sequence and molecular structure also support this scientific community of the scientists in understanding the potential threat and help and develop the preventive measures against this mortal disease. I would like to greet my colleague, my coworker, my team, Sherry Bachkat, Wolfgang, Daniel, Terry Mill, Robert Heyman, Slan, Kunza, and Noreen. These are my people. This is my team. They have supported me. They helped me to make my this presentation today. I acknowledge to Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery Technology Development Incorporation Canada. And at the last, I'm very thankful for staying with me and watching the video today. Thank you very much. See you soon. Take care. Bye now.